living things were composed of cells. By 1831, microscopes had improved to the point where cells were seen to have a nucleus, and it was soon found that without a nucleus, a cell could not divide. The secret of reproduction was clearly in the nucleus, but unfortunately, since cells are transparent, no one could see what was inside it. A hint of how the nucleus might be working was discovered by an Austrian monk named Gregor Mendel. Mendel studied the reproduction of pea plants in the monastery garden. He worked with purebred plants, one strain of which produced smooth seeds, the other wrinkled seeds, and he cross-pollinated them by hand. Crossing the two types of seeds always bred to a second generation of all smooth seeds. But when these were crossed with themselves, the third generation were roughly 3/4 smooth and 1/4 wrinkled. Why? By working some basic mathematics, Mendel concluded that his plants must carry two factors for seed shape, one for smooth and one for wrinkled, and that one factor from each parent went into the new seed. He saw that if the new plants received even just one smooth factor, all their seed would be smooth. In other words, the smooth factor was dominant over the wrinkled. But the next generation showed how there was a chance two wrinkled factors could recombine to make new wrinkled seed. Seed shape was just one characteristic Mendel tried. Others, such as stem length and pod shape, gave similar results. Two factors determined each breeding characteristic, and just one factor was passed on from each. In 1879, the German biologist Walther Flemming found that with certain red dyes, he could stain the nucleus of cells. And he saw little granules, which he called chromatin because they held the colour. When cells divided, these granules gathered in the form of threads, which were given the name chromosomes. They duplicated into double strands joined in the middle. Then these lined up, then split equally into two new cells. So, in normal growth, each cell carried an exact copy of the chromosomes. But in forming sex cells, such as eggs and sperm, or the pollen of plants, the chromosomes divided differently. First, the double strands lined up in pairs, so that the daughter cells took different chromosomes, each getting half of them. Then they split again in the regular way, separating the double strands. So, sex cells carried only half the number of chromosomes of a normal cell, and they were not clones of each other because they carried different halves of the original chromosome pairs. Clearly, the way that chromosomes paired up before a sex cell divided explained the way that Mendel's two factors were transmitted. Different species carry different numbers of them, up to about 250 pairs. Humans have 23 pairs. Yet there must be thousands of factors, or genes as they were later called, to carry all the instructions needed to create an organism. Somehow the chromosomes must be the carriers of the genes. But how? And how did they create the effects that were visible in the offspring? The answer to this last question came first. Biologists came to believe that the genes somehow controlled the production of enzymes. Enzymes were known to be giant protein molecules which acted as chemical controllers. An organism's characteristics are controlled by its enzyme makeup. Everything from eye colour to the digestion of food has particular enzymes associated with it. So the hunt was on to link enzymes to genes, and that required research into the chromosomes. Chromosomes were found to be made of a chemical called deoxyribonucleic acid, or DNA for short. In 1953, the DNA molecule was shown to be a fantastic double helix in shape, with a ladder of molecular groups like rungs joining the two strands. The strands were alternating sugar and phosphate units, and the rungs were of only 4 different kinds: adenine, guanine, cytosine and thymine, called A, G, C and T. These linked across in pairs, and A to T, or T to A, and G to C or C to G were the only 4 ways that they could combine. But each DNA molecule was huge, with thousands of these linking pairs on it. Taken in groups of 3, named codons, they were shown to follow the same coding patterns as the amino acid chains that make up the proteins in enzymes. The whole system is enormously complex. For example, since there are 4 times 4 times 4, that's 64 possible codon variations, and only some 20 amino acids to code for, most amino acids have alternative DNA codes. The process by which the code is transferred to the rest of the cell involves other chemicals like messenger RNA, and there are feedback loops. But the bottom line is that a gene is a series of DNA codons, which indirectly creates a similar series of amino acids to make an enzyme, and the enzymes characterize the organism. So, if you follow the chemical code back to its starting point, the genes control everything. Thousands of genes make up each chromosome, and the chromosomes come in pairs, so each gene has a partner on another chromosome. But as Mendel found, the partner genes are not necessarily quite the same. What happens if genes are damaged? For example, energetic radiation can fracture big molecules like DNA. Well, there is a backup gene on the other chromosome, but if the code is changed, then the resulting amino acid lineup of some enzyme is also changed. And the effect of that might range from being unimportant right through to being deadly to the whole organism. It's worst for the young, because cells are still growing and dividing, and any damaged code is thus copied and continued. If the change is in a fertilized sex cell, all the cells of the offspring are affected, and it may be passed on from generation to generation following the Mendelian rules, unless of course it's fatal, as it often is. Very rarely though, it is good, bestowing some change for the better, a trait worth breeding, which takes us back to the beginning of this story. Far more frequent, though, are genetic disorders. This is a form of epilepsy, a rare kind where sufferers have nightmares and seizures in their sleep. Science is now on the threshold of being able to link symptoms like these to specific genes. In this case, it was recently achieved by a joint research team in Adelaide and Melbourne. Dr. Sam Berkovic.
Not all epilepsy is caused by genetic abnormalities. However, in the case of the family that we studied to make this breakthrough, it affected 27 members over 6 generations. Of the more severely affected members, some of them never had a sound night's sleep due to this problem of epilepsy occurring at night.
Okay, hi Ben, I'm just going to examine you. Just want you to relax.
Here in Melbourne, we began the study of this family. My colleague Dr. Ingrid Sheffer contacted over 100 members of this family and carefully interviewed them, and then went to personally examine them to determine which ones actually had the condition and which ones didn't. Identification of which individuals have the condition is absolutely essential, so that our molecular genetic colleagues can then track the gene throughout the generations and positively identify it.
With affected members of the family tree identified, samples of their DNA were taken and analysed by a team of genetic researchers in Adelaide.
I was part of the team working on the epilepsy project. The first thing we did in Adelaide to identify the gene was to determine its location in the chromosomes. We did this by testing DNA from members of the epilepsy family, and we found linkage to genetic markers on chromosome 20. Here we have a picture of a chromosome 20 pair and a schematic diagram showing the location of the gene. We went looking for information on what is known about these genes and what they do, and much of this information is now available on the internet. We found a group in Germany who are working on a gene on chromosome 20, which is known to control transmission of nerve impulses in the brain. Within a few weeks of working with them, we found that the members of the family who had epilepsy also had a mutation in this gene. We had suspected that problems with nerve transmission might have something to do with epilepsy, and here was a genetic link that was unmistakable.
Well, it doesn't mean that we found the cure for epilepsy, but up till this time, nobody had been able to identify the underlying basis of any genetic form of epilepsy. Many cases of epilepsy are due to genetic causes, and up till this time, the drugs that we'd had available for the treatment of epilepsy were discovered in almost a random fashion. This important finding now allows us to work towards a rational basis of the treatment of epilepsy in the future.
So, how do we get our looks? Genes. But genes are not just the answer to the question of our outward appearance. The understanding of genes promises to unveil our entire biology. While at the moment we understand selected sites on various chromosomes, the breakthrough has already been made, which will allow us to systematically type the whole human genetic makeup. The simple breeding programs of the past will look primitive compared to a new ability to manipulate life in the future. Genetics is one subject that is set to change our ideas deeply.